Hi, everyone. My name is Kate Milliken. I'm the founder of MyCounterpane, and I've been living with MS since 2006. And today is another MyCounterpane 101. Today, we are speaking with Dr. Ari Green, who is the medical director of the University of California, San Francisco Neuroimmunology and MS Center. He's also the founding co-director of their small molecule remyelination program. Additionally, Dr. Green is a Harry Weaver neuroscience scholar with the National MS Society, and he's the first Debbie and Andy Ratcliffe Professor of Neurology. He is a man with lots of creds. Dr. Green, it's really nice to see you again.
It's great to see you too, Kate.
And I'm really psyched to talk to you about the subject of remyelination, which in some ways has become a bit of a hot topic. And I figured that one way that we could start is by you kind of giving context on the whole idea of myelin repair in MS in general. I've had MS for over a decade, and when I started my MS journey, feelings about myelin repair were nil, not possible, an impossibility. And I'd love to kind of see how far that's come.
Sure thing. So what motivated us at UCSF, this is really a partnership between a number of us who are interested in this field, particularly my good friend and colleague Jonah Chan and I, is that we're really interested in why does some tissues repair really well? So, we have an expectation when we cut ourselves or when we break a bone that that's going to heal. And yet when we suffer other types of injury, like the kinds of injury that happens in MS, we have learned to accept the notion that that injury has some degree of permanence to it. And it's a little bit ironic or surprising that that's the case, in part because if you demyelinate the brain of a mouse or another animal, it actually recovers myelin pretty well. There really is the capacity for myelin repair in broad, generally and broadly across a lot of different species. And the issue is what is different about MS? And then the second issue is, are there ways we can, regardless of what's different about MS, are there ways we can just overcome whatever the limitation is that's stopping myelin repair from happening, and leap to trying to make therapies that can allow repair to happen?
So when you kind of had this realization, certainly about other species or having no issue with myelin regeneration, and then seeing in the face of MS you had an issue, at what point did that kind of become as a recent discovery? Has that been known for a long time?
No, that's been known for a very long time. There are ways in which you can use a chemical demyelinating substance that is injected into the brain or fed to an animal and will lead to demyelination. And the reason many of those models are not great models for MS or have been thought of as not great models for MS is because the myelin recovery happens spontaneously. Within 4 weeks, if you digest the myelin using a soapy substance known as lysolecithin, if we inject that into the brain of a mouse and cause small lesions, those lesions will fully remyelinate within 4 weeks. And so in some ways, people thought of that as a challenge, and we thought of that more as an opportunity. We said, well, what do we know about that? And then, I really push it as a clinician. A lot of scientists in the field had thought about this for a long time, but the issue was in some ways, the scientific community doesn't have a way to think about, well, how is this exactly relevant for patients? And that's the essence of the partnership between Dr. Chan and myself, is to say, how do we bring different perspectives on this and bring it to bear on focusing on a challenging problem that we need to fix, which is myelin repair possible in MS? And if it is possible, can we use it to help prevent degeneration and clinical decline and reverse the ravages of MS, or prevent the kinds of complications that MS creates over the long term?
So was injecting those mice who had MS and actually seeing results that were positive with remyelination, was that the beginning of this study that you guys are working on now?
No, that was well known in the field, but what was really the genesis for what we did was having conversations in which the myelin biologists taught us in the clinical science and translational science community about these cells that are sitting there, kind of coming back to that first question I raised, which is why do some tissues repair? They repair because there are stem cells there, essentially, right? We sometimes think of stem cells as a miracle therapeutic that we're going to take from some other source and give them to people with degenerative conditions. Particularly, I think we're really interested in neurodegenerative conditions. But they're there. We have stem cells in our body. Those stem cells are what allow our tissues to repair themselves. And the thing that has been known now for a number of decades is that those stem cells for myelin are also there in the brain. And it's just that question of why don't they do their work? Why don't they mature and advance into myelin-producing cells and repair lesions after they happen? That's the crucial, central question, and one of the questions that, in some essence, we wanted to leapfrog and say, can we also, even if we can't exactly figure out why they don't repair, can we find ways to encourage them to do what they are supposed to do and make new myelin? And so that is the real central starting point of where our research began. And then Jonah made some amazing discoveries and some really amazing scientific breakthroughs by working out ways in which we could test large numbers of medicines, large numbers of either undiscovered chemicals or medical therapeutics that might be used for other purposes to see, could they cause those stem cells, those, we call them oligodendrocyte precursor cells, could we get those oligodendrocyte precursor cells to turn into oligodendrocytes and make new myelin, and do that in a way that was quick and was, as we refer to it as a drug screen, use it as a way to identify medications quickly and robustly and in a way that wouldn't take years and years, but might be done in much shorter periods of time.
So what's pretty cool about this whole idea is I think people have this preconceived notions, that scientists are always looking for the why, right? To really dig into the technical and really understand everything on a clinical level. And if you talk about the fact of understanding that stem cells aren't working properly as a 1, and you want stem cells to be working in MS as a 3, it's almost as if you guys are skipping the 2. In terms of the actual technicality. What is the motive for that? Right.
So, I mean, it's not that we're not interested in the why, and we think the why could be centrally important to figuring out future problems, but we also recognize there's an urgency here. I mean, I'm a clinician. I'm with patients all the time, and Jonah he's a scientist, first and foremost, but he's also a person who is, he's heavily motivated by trying to do good work. And so he's the kind of guy who, when he meets patients who ask him questions that kind of challenge some of our scientific assumptions, he says, well, what can we do now that might make a difference? And that's what kind of brought the two of us together. It's what motivates us to say, let's try and be a little disruptive, do things immediately that can answer some questions, recognize that there are limits to that approach, and not become hamstrung by trying to always just reach for the breakthrough. We recognize that there's laborious work that's going to have to go into moving this field forward. There's a lot of people involved. It's not just the two of us. There's a broad community of myelin biologists who are going to help push things forward. But we also really want to push the envelope and say, again, what can be done immediately, and how can we use what we learn that's done immediately to help, again, advance the field? That's sometimes referred to as a bench to bedside and then bedside back to bench approach. For us, it's really pioneered by our colleague and mentor, Steve Hauser, who that's really what's driven his career. And he pushed us to say, again, this question of, sometimes we think you advance from the test tube to the animal model to
phase 1 to the
phase 1, phase 2. Exactly. And we said, that's really not the way real clinical science works. In cancer, that's not the way things have worked at all. In many ways, the cancer field has been tremendously disrupted in a good way by people who said, let's check out what we can do for people who are, in situations that are desperate, that can't wait. And we recognize that inability to we can't sit on our hands permanently trying to figure out the whys. We've got to get to the hows.
Well, what's so great from a patient perspective and representing the patient population is, having heard about the International Progressive Alliance, having heard about all this incredible science that's coming down the pike, there's always that last, the last line of the interview of how long is it going to take? And somebody will say, oh, it's such exciting research and we'll be able to get the results by 2018, right?
Right.
So just to give an example to people, in terms of Dr. Chan and you looking at different therapies that already are in existence to help with this regeneration problem or fix it, what is the time that's saved?
Sure. So, Jonah made some discoveries probably 6 or 7 years ago that he didn't think were going to really lead to therapeutics, but he recognized that you could cause these precursor cells, these stem cells, to wrap even inert substances. So you didn't have to have a nerve cell for them to wrap. And but in 2013, so just a few years ago, he came up with an idea to set up a screening method. And the miraculous thing is, after he developed that, he sent off the prototype to an old semiconductor factory down in Texas. And they actually made the prototype within a few weeks. And within a few weeks after that, we'd screened all the available drugs ever approved by the FDA to see, could they cause these oligodendrocyte precursor cells to turn into oligodendrocytes? And we came up with a bunch of hits.
Wow.
And that was amazingly exciting to us. And then we said, well, are there any of those drugs that we are safe enough for us to try with people? And we figured out that one of them really looked like it was a great candidate. We pulled all the records from the FDA, contacted them. Again, sometimes I think in patient communities, understandably, regulators are seen a little bit, maybe not as the enemy, but as a barrier to getting treatments forward.
Which they are, although they do it with the right intentions, right?
Exactly. No, exactly right. These are people with an important job to do, which is keep all of us safe and keep the field marching forward. But they do have a tendency to sometimes restrain movement forward. And the neat thing was the regulators in this instance gave us all their records from a drug that had been in existence since the early 1970s. We looked at everything and saw this drug looked like it had a great safety record. It had some drawbacks. It probably causes some fatigue. It's not a drug made for the purpose that we're trying to use it for. Mm-hmm. But we thought, could we do it? Could we try a clinical trial in which we gave this medication to people? And it's an antihistamine. And so it had been used for years and years before the new generation of non-drowsy antihistamines have been developed in the 90s. And so we decided to test it out, and that led us to a clinical trial. Now, the other cool part here is that philanthropy and support, is a kind of advocacy that comes from a motivated community of people who want to see these problems solved, also help push us forward. They pushed us to say, when we said, well, we're going to have to go through normal funding mechanisms and figure out some of these things, it'll take a few years to get responses. And that whole process is valuable. You get feedback, but it's delayed. And so they said to us, can you do something impactful and important now? And we said, sure, if we had enough money. They said, how much money would you need? And they wrote us a check for that money, and we did the research. And the trial was done within 2 years and was completed by early this year. And the results we presented at the American Academy of Neurology. And it's still an ongoing process. We're even continuing to look at the data that we saw. The results were successful. The results suggest that there is a remyelinating effect for the medication, but we don't know how long is the optimal time to give it. How much do you have to give? What's the optimal dose? There's a lot of stuff that still needs to be worked out. And then it's, could we tune a better drug? Could we make a medicine that was really targeting this specific molecular keyhole
that didn't have fatigue?
That didn't have fatigue as a side effect. That that was just tuned for this purpose.
Right.
Can we do that? And so we partnered with a couple pharmaceutical chemists who've had a track record of making successful drugs. They actually have brought a number of drugs to market, and they jumped at the opportunity. They thought, they recognized, I think broadly in pharmaceutical chemistry, people have said, why can't they make drugs that work in neurodegenerative diseases? What's the limitation? What's the stop?
Yeah.
And they were so excited at the opportunity to work on this. And they've really been, this is a group from down in San Diego, known as Inception Sciences. They have been amazing partners at trying to help us develop a new generation of drugs and figured this whole problem out and navigate this whole process that, I'm a simple clinician. Joan is a basic scientist. It's going to take a broad team of people, a lot of folks, and probably a broad community of patients to get us drugs that are going to work in a concerted manner. But we hope to if we can bring anything to the table, it's urgency. It's the need to push that forward as fast as we can and as safely as we can.
And if you had a best case scenario, not holding you to it in any way, shape, or form, where do you think that lands as if it was ever able to really come out as an MS drug?
Yeah, so I think and I don't mean to hedge here. It's a hard question to answer because we don't exactly. We're still trying to work out how do we best test this. Now we have a little bit of a pathway set for us from this first trial that shows us we can do trials that are phase 2 that are proof of concept relatively quickly. How long will a phase 3 trial go? That is going to be up to a discussion between us and patients and regulators. And I think patients, have that combined tension, which they should, of impatience, which is to say, we want this now, but also to say, we want to know that this actually works. We don't want false promises. And doing something that's too quick could lead to false promises.
And a better way maybe to think about it is that if you hadn't taken this already existing drug, we would have already added 2, 3, 4, 5 years to the process. Is that fair?
You're right. Exactly right. And then the next drug, do we want to do a trial that's going to lead to approval of a drug? Should that trial be 2 years, 3 years, 4 years? Those trials no one's done a successful trial at the phase 3 regulatory stage ever in anything having to do with neurodegeneration. And so in some sense, the field is wide open, and it really needs to be a conversation, as it was in the HIV community, and it's been in the cancer community, between patients and regulators and doctors to say, this is what we expect, this is what we need, this is what we want, and really use that as a way to make a program that's going to really make for breakthroughs, meaningful breakthroughs.
Last question for you, which is, I feel, again, from the patient perspective, that the world of MS patients wondering what they can do now. What's really cool about this conversation is hearing a scientific team having that exact same mentality. And on some level, whether it's me going to my neurologist and saying, look, I really want to know about my diet. Is it going to help with my MS, right? What can I do now? Is pushing the envelope of a clinician to have to answer that question. I feel like in light of what you're doing with your study, you're also pushing the envelope of kind of the bigger foundation of the way it's been. So do you feel on a bigger level as a result of this that there's kind of a tectonic plate shifting on, your level of MS? Because certainly for me, watching a more holistic approach come into focus, I feel a tectonic shift happening in the way that MS is being treated. Any last thoughts on that?
Yeah, I think that's really, really insightful. And there's a lot of forces that are driving that. One is we have really moved past the kind of patriarchal view of medical care. Medical care is a partnership between doctors and patients, and it ought to be about thinking about how does a patient live their best life with the advice and guidance from their physician, but not the permission, right? It really is a process of interaction. And that's why it has to be about a really complete view of things. Medicine plus, is there lifestyle modification that can make a difference? If there is one thing I can really encourage amongst patients is the thing that's really clear, and it doesn't need all that much evidence to help motivate us, is to say activity is super important. I can't put my exact finger or tell you exactly what type of activity is great, but I can tell you broadly that physical activity, mental activity, social activity, those are the keys to a life well lived, and a life well lived with MS, because it has probably some significant reparative or at least compensatory effects on the brain. It allows us to overcome things that we would have thought, a few decades ago, the way in which physicians advise their patients was to say, well, there's not much you can do about this.
Right,
And
go rest.
That's wrong.
Right.
Exactly. And I really think that's wrong. It's the exact opposite. It's get out there, be active, be engaged in the MS community if that's valuable to you, or be engaged just broadly in the world, because that's going to make, that is a way to fight MS. It is a way to beat back whatever sentence MS wants to try and impose on you.
Yeah, that's awesome. Well, Dr. Green, I'm super psyched to hear what happens. And again, it's really cool to feel that things are moving faster and faster. So it's been a real pleasure to speak with you. And thank you for joining us. Cheers.
It's such a pleasure to talk to you. Thanks, Kate.
Thanks.